E agora vamos dar início, então, à mesa redonda. Eu gostaria de chamar para compor a mesa redonda.
We are starting the roundtable, and for the roundtable, I'd like to invite Dr. Valadao, Erico, Freitas, and Matthew Harrington. Welcome, all. And the discussion is open about bioengineering, biological material. Good morning.
How should we start?
Yes.
Yeah.
I think there are some questions from the audience, and questions for your very nice talk, your excellent talk.
Thank you. Yeah. Okay. I see the question. So should I just go through the questions, or?
Thank you.
As you wish.
Yeah, I will try to organize some of the questions, and I will ask you some of them. I've seen that you also select some of them that you wish to answer. And yes, and first of all, thank you very much for being with us, and it's a good pleasure for us to have you here in this forum.
It's a pleasure to be here. I mean, I wish I could have made the trip down to Brazil. This would have been really nice. It's about -10 degrees out here, so I'm sure it's much nicer weather down there than it is here. But I'm glad to be here, anyways.
Okay, thank you very much. And yeah, maybe. Would you mind if I start with the question to Professor Antonio Valadao?
That's totally fine.
Okay, thank you. Amanda, okay, I will. So the question was written in Spanish. I'm not good at Spanish, but I will read it in Portuguese. So, Professor Valadao.
Professor Valadao, what is the role of the temperature of the water in the shell formation in the initial phases of the golden mussel?
É uma questão
It is a question, very interesting question. I do not know the answer right away. I do not know, because the golden mussel lives in the temperature where he is, where the water is. So it could be the temperature of ocean water, also fresh water. In our case, is fresh water temperature. I imagine the first observation, there is no influence about the water temperature and the layers produced. What we see is the phases. It's important is observing the phases that are the stages in the shelf layers are different. They do not depend on species. This is an important point. But I believe that the temperature variation in the environment do not impact the layer formation.
Thank you very much for your answer. And the next question, I think both of you can jump in try to answer. Yeah, it's written in Portuguese, and I will read it out in Portuguese. Existem alterações.
Is there changes in the bisu in animals that are exposed to stress, environment with the stress?
Professor Matthew, you are the authority here, please.
So yes. So when I was doing my PhD, I was at Santa Barbara, where we had seawater coming from the ocean. So the lab, I was lucky, it was right on the water, and we had seawater that was flowing into the lab, and we grew our mussels inside a tank with real seawater. Okay. When I started my group in Berlin, or Potsdam, Germany, it's landlocked. There's no oceans anywhere close, so we had to use a tank. And it was clear that the threads that were produced by these mussels were inferior. The byssal threads were inferior compared to the ones that came from the ocean in terms of mechanics. And, you know, we tried to balance the composition of the seawater as much as possible. We tried to match the right temperature and everything. But I think this environment is stressful for the mussels, and they simply don't produce threads as well. We see the same thing with the freshwater mussels that we look at. We take them out of the river here in Montreal. And when they get to the lab, they may produce threads for a few days afterwards, but they feel stressed in this environment that we bring them in the lab. So I think for sure, the local environment affects the threads that they make. Yeah, for sure. Yeah, the answer is yes.
Let me ask you a question connected to the previous one, if I may. Do you believe that the non-production of these byssal threads, the vesicles, because of the vesicles on the foot, or during the secretion of the proteins for the byssal formation?
Okay. So if I understand the question, you're wondering at what stage does this process, does the stress affect we don't know, but I could imagine that it potentially happens at both stages, but I would say probably more importantly is the vesicle stage, the packaging stage. Because mussels, you know, they take a lot of their energy to make the byssus. I forget the exact number, but I want to say the energy economy, something like 30%. But if they're reproducing or I don't know, they might not be able to produce the proteins in the same way. They might be not as fit nutrition wise, and they may be affected about the proteins they can make. So the proteins might be affected. The other thing is the acquisition of metal ions. So we've seen that metal ions such as iron, vanadium, zinc, copper are important for the formation process. If the mussel cannot acquire these metals in sufficient amounts, you might also have an effect on your byssal thread. So this is happening before the formation process. There was also some really interesting work by Emily Carrington where they showed a seasonal dependence on the byssus strength and stiffness, right? So they found that, in different seasons, the threads actually have different mechanics, and they never really understood what caused that. I think everything's happening beforehand, to be honest. I think the formation process, maybe it can be interrupted a little bit, but it's really probably more the health and happiness of the mussel, right? That it's getting the nutrients it needs and that it's able to produce the material, which is based on protein, right? If you're not able to produce also in your own body the proteins that you need to do the job, you're not going to be healthy. And it's the same with the mussels, I think. Thanks.
Yeah, thank you very much.
A forma como sua linha de pesquisa aprofunda.
It's interesting, your research deep dive on the byssal foot morphology. We have some more questions here. Let me see. Q&A, we have some questions. Let me read them. Professor Valadao. Dear Professor, very interesting, the layers of the shelf, especially the liquid crystal. The environment conditions of some elements, availability, carbonate and bicarbonate, may influence different ways the formation of the thickness of different layers, and what is the impact for the animal's life? I think that the same as Professor Matthew explained, the conditions, the environmental conditions really influence the formation of the shell as well. There is in my lecture, I showed you that is a point in time the meso loses control. So this layer is replaced by aragonite, and they replaced by another layer. But there is a morphology that is different. It's made of columns. There are some authors that say that this is the point in time where there's no enough food, and the meso is growing the shell, but without enough food. So this layer is affected. So we showed that this layer is filled with porous. There is porosity. So if the environmental conditions impact, and we say that the mussel shell is fragile, especially when we grow them in the lab.
Got another question for Professor Matthew Harrington. It's from anonymous attendee. Dear Harrington, Dr. Harrington, about the coordinate metal bonds in dopa metal, have you tested it with iron 2 ions or the reduced ionic species? So do you think does that metal species make any difference in properties such as stiffness or hardness?
It's an excellent question. We haven't tried with iron 2. Other studies on, let's say, bio-inspired hydrogels that are based on similar chemistry do show differences between the mechanics of a reduced iron or also vanadium in different forms. So this definitely can have an effect. It's just that nobody's tested it. One interesting observation, we've been really diving deep into the vanadium literature to try to understand how this is used. And it does seem like no matter, so at least with vanadium, the dominant form is the 5-oxidation state. But we found in the literature at least, that when this is coordinated with dopa or catechol, it's reduced to the V4 vanadilyl ion, and this is really interesting. And we do detect with EPR V4 in our threads. So V4 is very unstable at basic pH. It usually only is observed at acidic pH. We see it present in our threads at basic pH, and we think this really has to do with the fact that it is complexed with the catechols. So this is something we're really interested in the effect of oxidation state and actually how the mussel might control oxidation state. So yeah, I can't answer your question about iron 2, but I can tell you for sure that's going to have a big effect on mechanics. And it's not clear how this is controlled by the mussel.
Thank you. Another question from Professor Assis, our coordinator. So congratulations for your conference. So his question is related to the vanadium concentration in ppm or in percentage that you have measured. Yep. So that's the one question. The second question is related to the effect of the vanadium on hardness of the mussel.
Yep.
So did you measure the hardness? If so, please give us some figures, or maybe, yeah.
Yeah, so the first question, I mean, I can't give you an exact percentage. What I can say is that we can detect it with TEM EDX, but we cannot detect it with SEM EDX. And these have about an order of magnitude difference in their sensitivity. And talking with the technician on the machine, I believe the TEM EDX was sensitive down to 0.1 weight percent, and then SEM EDX was down to 1 dry weight percent. So it may be somewhere in that range in terms of weight percent, dry weight percent. It's not a lot of metal, but it is important. It does a lot. One thing that I didn't tell, and I think I saw some of the other questions were kind of asking about this, like how did they accumulate? We have some unpublished stuff that we're writing up right now where we have found iron and vanadium stored inside the mussel foot, and we've shown that it's being used to build the byssus. So in this case, they're stored in these highly concentrated little vesicles, where they are actually already complexed with catechol. So this is kind of an interesting finding that we made recently, that this is an organism now that actively uptakes, accumulates, and then concentrates vanadium ions. There's only three other organisms on the planet, or three other types of animals on the planet that have been shown to do this. Ascidians, one species of mushroom, and there's certain types of polychaetes, so worms that do this. Ascidians, so tunicates are one species that is well known for this, but nobody knows why they do it. So I think this question of vanadium is really cool because it's a really clear example that the mussel is actively taking it up, accumulating it, and then using it to make a hard material. In terms of the hardness, we measured values with nanoindentation of about 40 megapascal hardness, okay, and about 1 gigapascal stiffness. Now, nanoindentation is kind of quasi-static. That means we're not taking into account the dynamic nature of these crosslinks. These metal coordination bonds are important because they are dynamic. The bonds are breaking and reforming at a certain rate. They have a certain dissociation rate, constant. This determines their viscoelastic properties, which I think are way more important than the hardness or the stiffness. And in that case, so what you may have seen from the talk is that the iron and vanadium threads didn't have a big difference in hardness or stiffness, but they will have a big difference in their rheological properties, in their viscoelastic properties, because these two bonds have different dissociation rates. It means that the bonds have a different lifetime. And it means that vanadium is going to be a longer lasting bond, and it's going to make a more solid-like behavior, whereas the iron is going to give a more viscous-like behavior, if you know anything about viscoelastic properties. These are two different types of bonds. By simply changing the metal ion, you can completely change the viscoelastic properties. It's just really hard to measure these properties at such a small length scale, so we don't have that data. But I'm pretty certain that that's the key difference between iron and vanadium.
I wanted to jump in. Yeah. Thank you. I wanted to jump in that interesting question because so we've mentioned that the mussels, they capture iron and vanadium ions and other ions from the environment. Yeah. So I suppose maybe, please correct me if I'm wrong. I suppose they capture those ionic species through the gills, or this is why I wanted to ask you. I wanted to ask you a question. Have you ever tried SEM EDX on your? Because you showed these really nice nanoparticulate, net particle or microparticles or nanoparticles that were sitting on the gills. Do you think that this is, have you ever looked at the composition of those? Do you see the presence of, metal ions that are present in these? No, we haven't looked at yet. So that's something that is worth it. And because as you told, those species in dopa metal bond, they are not. You mean in the vesicles? You mentioned that those are not nanoparticles, so they are only ionic species spread out in the vesicles.
Well, and we don't know yet. We don't know yet. All we can see, these things are submicron size particles. But what we do see is that there's a high organic content, and we see the presence of catechol metal interactions, which tell us that the metals are interacting with catechols. But also you have something like ferritin, which is, it's like a it's like an amorphous nanoparticle inside a protein.
Yes.
So that's also possible, right? So I can't say anything about that, but I am going to be investigating that topic definitely, because I think it's super interesting.
Because if these ionic species are not, we still don't know, but maybe I'm just wondering whether the mussel can dissociate those metals after capture those particles from the environment, and then they can be storage those metals somewhere.
You're spot on. So there was a recent paper from the group of Jonathan Wilker. I don't know if you know him, but he was so like it's desirable that if you want to study the metals in the threads, that you can put them in seawater with more metals. But if you put them in seawater with too many metals, like dissolved iron, well, first of all, iron doesn't dissolve very well, but if you put it too high, it kills the mussels. We tried this, it didn't work. So what he did was he just actually took elemental iron, like little nanoparticles of iron. The mussels acquired the nanoparticles and somehow broke it down and then used it to build their byssus. So it is possible. No, I mean, I don't know how it works, but it's a super interesting question, I think. But yeah, that's why I thought I found your talk really cool. Thinking about these are the questions we're kind of like thinking about. It's like, how are they getting these? Where are they coming from? Is it from food? Is it dissolved in the water? Is it particulate? Is it dissolved? Who knows, right? But it's very cool stuff.
Yeah, thank you. Another question for you. Let me see. That's done. Somebody's thanking you for the great talk and extremely interested in your line of research. In general, in mussels, we find a center of the byssus made up of collagen. It is the first time that I have heard of other materials for the central part. Do you have an explanation, even an initial one, for this interesting phenomenon in the case of the zebra mussel?
Yeah. So the mytilid species, and that also includes like Perna, so Mytilus, Perna, and a couple other species, they all use collagen. That's pretty well established. But actually, there was already, before we started this zebra mussel work, there was one other case of the giant clam. So that's Tridacna. These are these massive, clams that live in the Pacific, Asia Pacific, and they use alpha helices. So this was work done back, I want to say in 2008. They showed that there's alpha helices there. Then we started doing a bit of work on Pinna nobilis and Atrina, and also there's a oyster called Pinctada. They all belong to this group of mussels that are called Ostrida. So there's the mytilids, there's Ostrida. Ostrida mussels do not use collagen. Instead, they use globular proteins. So these proteins don't have any specific secondary structure, but the globules themselves organize into a superhelix like myosin. It's not the same as myosin, but we kind of solved this structure using small angle X-ray scattering. So that was a huge eye opener for me. I felt the same way that, well, this should be collagen, but it's not. Then we started studying the zebra mussel, and we discovered immediately this is a beta sheet. It's almost like silk. It has a similar structure to what you would find in spider silk. So the thing I take from this is that to add to that, the mechanics of these four different types of threads are not that different. That was what really surprised me. But what I really think we have is an example of convergent evolution. Convergent evolution. It means that this was independently evolved at least 4 different times, and using different proteins that were kind of just laying around, like collagen. Obviously the mussel is using collagen because we all vertebrates are using collagen, and I don't know. So I think the point is that these threads are useful because they allow mussels to attach or to anchor, And so they've evolved separately at least 4 different times to result in something that has similar properties. This isn't that crazy because silks, what we call silks, like spider silk, silkworm silk, then there's also all sorts of insects that are making things that are similar to silk, but some of these are based on collagen. Some of them are based on alpha helices too. So it's not that crazy of an idea. It's just really cool to study this and try to understand, why and how these all evolved. And I'm sure there are other examples out there that are even different from these 4 examples that we know about now. So yeah, I agree. It is kind of a shocking observation and very strange. But the more I investigate this, the less surprised I am to find these new things. But it's a great question. Thank you.
Yeah, I have a question for my own. And you've mentioned that the muscle, at least the medialis agilis, so it demands 30% of energy when they are when they're making the bases, right?
I pulled that number out of my memory, which might not be as good as it once was, but I know it's high. It's double digit. So, it could be like 10, 18%, 30, I don't know.
Oh, wow.
But they use a significant amount of energy towards making these bissel threads.
Yeah, I was wondering if whether the muscle keeps its valve open or closed while during the bases, because also they spend a lot of energy when filtering particles. So I'm just wondering whether they can do both tasks at the same time, or they.
That's a great question. I mean, when a mussel is making a byssus, I don't remember seeing like the mantle hanging out or anything like that. It's in a very high, it's in a tense state, right? So it has this hard shell to protect its body from being eaten. This is the one part of its life cycle where it has to put its soft part outside in the open. And so even the slightest movement of my hand in front of the tank, it pulls it in. And that means somehow it has photoreceptors that can detect maybe shadows moving or something like this. But I can't ever remember at the same time seeing it actively filtering. But yeah, I don't know the answer to that. But it is a high tense state where, and it happens really when you remove the old byssus, we put them in our tank. Over the next 24 hours, the mussel will get busy making 20, 30, 40 threads really, really quickly, and then it won't do anything for a while. So it's like a lot of energy at once, and then nothing. Once it makes that attachment, then it kind of chills out.
Thank you. So another question for you on. So it's about excellent. Someone is saying there was an excellent lecture, and separation of amorphous phase and spinodal decomposition are well-known theme in science over inorganic gases. So do you evaluate any similarity with what happens in vesicles that are presented?
Yeah, no. So that's a fantastic question. So I'm less knowledgeable about these inorganic phases, but I understand the analogy and the connection. This sort of phase separation and ripening that we observe in these structures, it's definitely a physically driven phenomenon. We draw the analogy closer to not so much the amorphous phases, but of inorganic phases, but more these block copolymers. In which case you do have this kind of phase separation, based on the fact that you're mixing two polymeric protein components that are immiscible. So you're confining these two immiscible liquid phase proteins in an environment. You're concentrating them, but they don't mix because we think this has a lot more to do with having segments that are non-polar versus polar, right? Polar and nonpolar don't mix. So they instead phase separate. And depending on the lengths of the different domains, the nonpolar and polar domains, they'll phase separate into different textures. And that one of those textures that we observe is that gyroid-like texture. The really interesting thing here is that different mussel species have different textures, but they all resemble different like block copolymer phase separation structures. So this would suggest that based on the combination of nonpolar and polar lengths, or the relative concentration, you can get these different phase structures. So that's what I think. And yeah, this phase separation stuff is also kind of a new literature to me, so I'm still diving into it, but I find it super interesting, and I'll definitely look into these this idea of inorganic glasses. I hadn't come across that yet. So thanks for the great question.
So Professor Valadao, you were in mute.
Professor Matthew.
Professor Matthew, I asked you these questions about glasses, inorganic glasses, because it's important to us. When I mentioned about crystal liquid, I was based in your article idea. I think that you were the first one mentioning the liquid crystal aspect for the Bisu formation. But I was thinking about it because in the spinal decomposition, you have water in the glass. What I mean is, when somebody's taking a shower, the water that's growing, there are two phases and they are separate. That's the decomposition, spinal decomposition. It may happen because of energy, low energy effect. We called high organization. So I ask myself, in order to advance in our research, what would be liquid crystal and the spinal decomposition or any other, because there is this decomposition that is instantaneous, and there is the growth and nucleation decomposition. So I'd like to hear from you, because I think that is so interesting when you mention the phase separation.
Yeah. So, I mean, I have to say right off the top, I am not an expert on spinal decomposition or these sorts of things, but what we think there is probably a maturation process in the mixing of these two proteins. So what we're looking at when we see that phase that you see, this gyroid phase, where you have this phase separation, is kind of like the final form. But during the process of mixing these two proteins together and concentrating them, probably you have various stages of this spinodal decomposition. Another example that we're studying right now, which is really interesting, I didn't present it yet because it's not quite there, but we're also studying the formation of the adhesive. So in the talk, I showed you this picture of the plaque is the adhesive, and it has this porous structure. The proteins are stored inside vesicles, and it's just a condensed liquid phase, so there's no porosity. But what we've found now, we can purify these vesicles, and then we can do in vitro studies on them. We pop the vesicle. You are exposing now this condensed liquid protein phase to water. The water moves into it and forms nanodroplets that then over time mature, they mix together, but then at the same time, the proteins are cross-linking and hardening. So this then locks in the porous structure of the protein. So I think maybe this is even closer to your kind of spinodal decomposition, in the sense that in this initial stage, there's no water. Well, it's highly concentrated. There's not a lot of water in there. You open it up, the water moves in, but it doesn't mix, right? It doesn't mix with the proteins, or maybe it doesn't even move in. Maybe it's already there and it just phase separates. But then you have these little droplets, they're nanoscale droplets that then fuse together and form larger droplets. So I guess this would be closer to like Ostwald ripening or spinodal decomposition. And then they're hardening at the same time. So it's a kinetic problem of how quickly do the water droplets mix and how quickly does the crosslink formation happen. And we found we can control this rate with pH. So we can slow it down with low pH, we can speed it up with high pH, and then we get different morphologies depending on this. And this is actually, we discovered after the fact, this is a technique they use in 3D printing of porous structures. So the work of, I don't know if you're familiar with Andre Studart at ETH in Zurich, but that's what they do. They do 3D printing where they have a chemical reaction that leads to, they have a light initiated phase separation, but at the same time, there's a chemical reaction that's cross-linking this. And so by controlling the intensity of light, they control the porosity of the final product. So I think there are definitely there's some definitely really interesting points that you're bringing up on this formation process. And we're only at the beginning of really understanding what are the physical and chemical driving forces of solving this problem.
Sim, mas a linha de pesquisa.
But the line of research that you develop is extremely interesting because we try to understand, we believe that is a long-term process, trying to understand how to produce material that are sustainable, materials with sustainability, materials that we are made of, our body is made of. I believe this is a central point. I would like to conclude the spinal decomposition. Dr. Eric comment about energy, and this is an important point for spinal decomposition. The energetic cost is low, an important point. Maybe you can study about viscosity, because this decomposition happens when the viscosity is different.
Yeah, no, that's a great question. So we just started, so in terms of viscosity, we started a collaboration with a group that does optical tweezer-based microrheology. This is a technique where you take very tiny bead and you oscillate it in a light field. So optical tweezers are holding onto the bead, and then you apply an oscillating frequency on the bead, and then you measure how this varies inside the solution. And from that, you can pull out all sorts of viscoelastic properties, storage modulus, loss modulus, damping factor. And because these beads are so small, some of them are 80 nanometers, we're going to try to get them inside the vesicles and actually measure the viscoelastic properties inside the vesicles. And we'll be able to say something more intelligent about the viscosity. But I think you're spot on with this. You know, the other thing I didn't really mention is, again, you also have this place where the plaque, the adhesive, has the collagen that comes into it like this. And this is spontaneously formed. And I think this really has to do with differences in viscosity. And it's this thing probably you're aware of called a fingering instability, where you mix together two immiscible fluids that have different viscosity. You get this kind of fingering instability. I really think this is what's happening there. I just don't have the proof. But this is you're spot on that these methods of low energy spontaneous processes that occur could be a really cool way of building complexity into a material like this. The neat thing, well, yeah, the challenge is actually proving this and pulling the information out of the system. But it's a lot of fun to try to figure it out. But I think your questions are absolutely spot on. Thank you.
Thank you, Professor Harrington. We've got another question for you. I think you've somehow addressed that question before, but do you
This is the
believe this biomineralization
yeah.
of vanadium, so in the granules that also accumulates in the body?
We don't know yet. My feeling is that they're not, well, there's a lot of organic in there. So it could, I guess it could be some sort of mineral phase, but it's also incorporating quite a lot of organic, probably protein-based storage molecule.
So in those measurements you've done, the Raman spectroscopy, so have you found any evidence of in mineral phase, a vanadium mineral phase or iron mineral phase on that?
No. And, you know, no, we haven't, because, or at least I'm not aware of it, because the peaks that you typically get from proteins are very broad and poorly defined, right? Because they don't scatter as well as like crystalline solids. So typically, I mean, we've done some studies where we are measuring minerals, and the peaks are so much sharper and more better defined and more intense. And so I feel like if we had hit a mineral phase, we would probably observe it, and we've measured a lot of samples. That being said, of course, if you have an amorphous mineral phase, maybe it won't be quite as intense. But I have to say something about the Raman measurements that we make on these metal coordination bonds. The reason that those peaks are so intense is because this is a resonance Raman effect. It means that the metal complex actually is a chromophore that has a absorbance peak that's near the laser we're using to measure the spectrum. So if your absorbance peak of that metal complex is close to the laser, sometimes you get an enhancement of the intensity of the peaks. That's why they look so intense. It doesn't have to do so much with the concentration or the the relative abundance of protein versus metal. It really has to do with the fact that we have resonance effect that's enhancing these peaks. That's the only reason we're able to see them, probably over the fluorescence. So I can't say for certain that maybe there isn't a less abundant mineral phase there, but I haven't seen any evidence of it yet.
Yeah, I was wondering whether you could see something using maybe a double corrected TEM, doing electron energy loss spectroscopy. I was looking at the vanadium L edge, so it's quite close to the oxygen K edge, but maybe you make it harder, maybe, unless you use a very nice resolved image yield spectroscopy.
Yeah, so we just got that on campus here at McGill. So it's something that we do have planned to kind of check out. Also, I mean, probably our machine's not sensitive enough, but we want to look at XPS and try some of these other techniques. But yeah, you're absolutely right. Eels would be great to try out, but there might be some challenges there as well, like you mentioned.
Yeah, I suppose that. Thank you. Another question on Professor Harrington. Are those pisovesicles produced and secreted in a typical exocytic pathway? Are there cell membranes around them inside the muscle cell, or are they organized in a different way?
It's another great question. Thank you, Anna. So it is a typical exocrine secretion process pathway. They are stored with membranes. And actually we can extract the full intact vesicles out of the body of the muscle and keep them purified. So we basically grind up the muscle feet, and then we do a density centrifugation gradient, and we can extract just these vesicles. We can extract the ones that make the core, so the collagen vesicles. We can extract the ones that make the plaque. The ones that make the cuticle, the outer coating, are still hard. They're really small, and they seem to be more sensitive. So they definitely have a membrane. And what we can see is, so for example, with the core vesicles, we published a paper recently, or in 2019, where if you put them in a hypoosmotic environment, right? So in order to purify them, you have to have a really high concentration of sugars in the buffer, otherwise they pop. When you take them out of this sugar-rich buffer and then put them in just water, they burst. And if you put them in a slightly lower osmotic strength buffer, they'll slowly swell and expand. So you can actually see them changing shape because they have this kind of football shape. They'll expand till they become spherical almost. They lose their birefringence, so they're no longer in a liquid crystal phase. Because the liquid crystal phase is lyotropic, it means that it's concentration dependent. So as soon as you lower the concentration of the protein inside the vesicle, they change, right? So it's definitely membrane bound. Now, when they're secreted, what's not clear to me is what happens to the membrane. I think that the membrane gets removed as they're being secreted out. It gets peeled off, and the proteins are then expelled into this long groove. So it's kind of like a polymer injection molding or something like that. But yeah, they definitely have a membrane. It seems to be a typical kind of exocrine pathway. The cells that produce these store massive amounts of these vesicles. Like if you look at the cell, if you look at these TEM images of the cells, it's almost all these vesicles. You see the nucleus, you see, you know, some other things there, but it's almost all these vesicles just packed together, ready to go. But good question. Thank you.
Yeah, thank you very much. There are two more questions left. Would you mind to go through them? Or?
Sure, no problem. I'm happy.
Okay, thank you. Thank you for your excellent lecture, very rich and deep on the topic. According to you, the pH of seawater would assist in the process of organizing collagen molecules. But in the case of fresh water, we would not have this variation. The pH could vary little from the interior of the animal to fresh water. When then would produce this alignment of molecules to form an extremely resistant species in fresh water?
Yeah, that's another great question. So many great questions. It's all stuff that we're like interested in right now. So I would say, so obviously the inside of a cell is going to be around neutral, but we think that the local environment inside the vesicles is quite acidic. So this is a special microenvironment that the organism creates inside the vesicle. So we think that, you know, the collagen vesicles are probably around pH 5. The adhesion vesicles have been measured at pH 2. And so these are really acidic. So I don't think it's totally true to say that the freshwater mussels couldn't do the same thing and create a special microenvironment inside these vesicles, which we do see vesicles where the alpha helical proteins are stored in the zebra mussel and the quagga mussel. We just don't know much about them. We don't know what the pH is or anything like that. But the other point of your question, or the other point to address this question, is that the freshwater mussel is completely different. It doesn't use collagen. It doesn't use histidine. It apparently doesn't use metal coordination. We have no evidence of that yet. Instead, it uses this completely different approach of alpha helical proteins. So we think they're very long, rod-like proteins. It could be a similar liquid crystal type thing. We don't know this yet. But then you start off with an alpha helix, which is a coiled coil, which is a nice way of storing a protein in a kind of stable state. And then what you're doing is you're using mechanical force. So instead of the chemistry being so important here, it's really the mechanical force of stretching the alpha helices into beta sheets. Once you stretch them into these elongated forms, the neighboring protein chains then hydrogen bond to form a sheet. And our evidence tells us it goes beyond that. They form stacks of sheets called crystallites. That's what our X-ray diffraction tells us. So it's a completely different mechanism. that's interestingly more dependent on mechanically driven protein conformation changes than the chemistry driven metal coordination that we see in the middleist threads. I'm not saying that there isn't this also a pH dependent trigger. I don't know. We haven't measured that yet. But it's just a different material. It's a different processing pathway. And yeah, I don't know. Yeah, I don't know what the role of pH might be in that yet, but it's a great question. Thanks.
Thank you. The last question, and the induction of species formation use the same protocol, same procedure proposed decades ago.
Yeah.
Do you predict that some research in this direction, apparently simple, could help to understand the phenomenon of formation of species?
Yeah, that's a great question. So yeah, we're still using the same protocol from Tamarin from 1975 or 4. It's an old method, but it works. And but it almost works. It's like it's a little bit over the top. Half a molar potassium chloride injected into the foot, it causes this massive almost seizure in the muscle foot and the secretion of all these. It's probably a bit over the top and probably could be achieved in a more subtle way. We did at one point, I had a summer student who kind of looked into this a little bit to see if we could lower the concentration and or if we could stop the process with muscle relaxants like drugs, that stop muscle contraction, without any real big discoveries. But I totally think this would be a really cool area of research, and could be really interesting. Also, kind of from a neurological standpoint, looking at the nerve center, because the idea here, unproven, is that these ions are then activating this kind of nerve center that's leading to the contraction of these muscles. But in the original paper from Tamarin, there's no explanation for it. It's not even like discussed in terms of why they chose to use KCl, why they chose to use half a molar. They don't explain it at all. And I haven't been able to find, they don't cite any literature, and I haven't been able to find an earlier example of why they would be using this. So, whoever has asked this question, you're spot on. This would be a really cool area to try to understand the control of this. And if you could understand the control of this, you might be able to do this in a more subtle way that might result in something that looks more like a real Bissell thread. But yeah, I don't know. It's a great question. No, thanks.
Okay, thank you very much. And I'm particularly interested.
Temos mais alguma pergunta, Dr. Érico?
Is there a question, Dr. Erico?
Yes, but I'm particularly interested in just one thing. So one of your PhD students have done slice in view, feed milling slice in view,
Yeah.
Great results. So I have them trying to perform serial section array or the technique in the volume SEM scope to look at the larger structures.
Yeah, I mean, there's different length scales. So the thing we're really trying right now to get going is TEM tomography, which is an even smaller length scale. So I already think of the FIB-SEM as a massive length scale. But yeah, you're absolutely correct. It would be really interesting to look at this as kind of a machine, as a factory, and to understand really how the whole factory is connected and the role of the groove and everything. That's something we're definitely trying to figure out. To connect the different length scales to figure out how the whole process works, seeing it as kind of like a holistic complete process. But yeah, FIB-SEM is nice because it gives us a relatively large volume on the scale of tens of microns by tens of micron volumes, but still gives us still gives us like an overview, you know, still gives us the detail of what's going on inside the vesicle at the kind of nanometer length scale. So it goes from nanometer to micron. The TEM is going to just go from like, you know, 1 micron, you know, volume and with a thickness of maybe 200 nanometers. And then, but you get much higher spatial resolution at that length scale. The next length scale up that we've been doing is histology. And in that case, we're just staining the sections with different histological stains. And in our 2017 paper, we've done a serial section down the whole foot, where we look at where are the glands that you know, these different glands, where are the different proteins stored. So that is something we have done, but actually trying to get that length scale in between where you can get like a whole foot analysis, but then have kind of more nanoscale structural resolution, that's challenging. And we haven't quite figured out a way to get that yet, although there may be some ways of doing that. But yeah, it's a great question.
Thank you very much for your answers. I think
muito obrigado, Dr. Matthew.
Yeah.
Dr. Matthew, thank you so much, says Amanda. Dr. Valadao, you are so nice. He said, you are so nice. We thank you so much, but you are so nice, Professor.
Thank you.
You have given us your time, and we really appreciate it. And we hope that in different opportunities, we might have you here in Brazil.
It'd be nice.
Thank you, Matthew.
Yeah, someday I hope so. It was a real pleasure to chat with all of you, and it was really nice to also hear about your work. And I really appreciate the invitation. And I'm also really impressed with these amazing questions, because you really are like spot on the exciting aspects that we're interested in at the moment. So I appreciate the conversation. I got a lot out of it.
Thank you.
Thank you. Obrigado. Yeah.
Yes.
Obrigado.
Did I say it right?
Without accent.
Yeah. Obrigado. I try. Okay. Thank you.
See you. Bye.
Bye.
And if there is something that some
Obrigado,
words you
doutor.
could say at the end, so Amanda. Sorry. Feel free if you want to say some few words at the end of this today's forum, and if there's something you'd like to address or something you'd like to say, and yeah, feel free. It's okay. Thank you very much again, and hope see you next time, and see you soon, maybe.
See you. Bye.
Obrigado, Dr. Matthew. Estamos caminhando, então, para o final do nosso segundo dia do Fórum de Discussão. Gostaria de agradecer a todos que estiveram presentes. Só lembrando, então, que amanhã o nosso fórum começa à tarde, né, diferente de hoje, começa às 13 horas. E é isso aí, tenham todos uma boa tarde e amanhã a gente se vê de novo. Muito obrigada a todos pela presença.